Hi everyone, my name is Carter. Today I'll be talking about SDS PAGE in a step-by-step method using Rockland's protocol and also providing you like the theory and the practice of doing SDS PAGE. SDS PAGE stands for sodium dodecysulfate polyacrylamide gel electrophoresis. SDS is a detergent, and then PA, which is polyacrylamide, is the kind of gel that we'll be making. And then gel electrophoresis is the technique. That's what the entire acronym means. Make sure you check out SciGine.com. Make sure you like and subscribe to this channel because I'll be teaching you a lot of different biochemistry and biology methods. And you can find over 1 million free biology methods that you can actually use in lab at SciGine.com. Now let's get into it. So today we'll be using this protocol from Rockland Antibodies. These guys make a lot of different antibodies. They've done a lot of Western blots, SDS PAGE, etc. So it's a pretty reliable protocol to depend on. And this link, which is the actual protocol itself, I'll link to it in the description. So what is SDS PAGE? SDS PAGE is a method for separating out proteins, typically, by their size. And we've already talked about gel electrophoresis in the past, so I won't really get into the details of what gel electrophoresis is. But here you can see that we've got a gel, and inside the gel we've got our samples here. And each of these samples has now traveled over time so that distinct patterns emerge within the gel, which is all the different proteins that were in the sample that were at the top are now separated throughout the gel. So that's really the idea behind SDS PAGE. We're taking a mixed bag of proteins and then we're separating them by size. And we're doing that using electrophoresis, which is a technique where we apply an electric field. All the samples that are negatively charged go towards the positive electrode, and then all the samples that are positively charged go towards the negative electrode. But here, we've made sure that using SDS, we actually don't have any of those positive samples. We'll cover that later in this YouTube video. All right, so step 1 of the protocol is to decide which percentage of gel you need to separate out your proteins. And then they've given 2 examples. Use a 4 to 8% gel to separate out proteins between 100 and 500 kilodaltons in size. And then the second example is to use a 4 to 20% gel to separate out proteins between 10 to 200 kilodaltons in size. Wow, that's a lot of information. Let's break it down. So what does it mean to choose the percentage of your gel? To understand this, we first need to understand porosity. And I've covered this previously in the gel electrophoresis YouTube video, so I'll link that in the show notes. Let's just cover that quickly here. What porosity means is that within our gel, you can actually take a cross-section, which is this thing in gray. And when you take the cross-section, you'll actually see a whole bunch of pores. You'll see some bigger pores, and you'll see some smaller pores. When you have a sample, like a protein, that you put inside the gel, that protein can transfer through those pores. If the pore is big enough, like this one here, then the protein will actually go through without any issues. If the pore is too small, that'll retard the protein. So that's what porosity means. And you can actually change the porosity of a gel by choosing the percentage of your polymers that are in there. So remember that in SDS PAGE, we're actually making a gel and we're using polyacrylamide. Generally, you take polyacrylamide and then you polymerize it using TMED and bisacrylamide, which is a crosslinker. So depending on how much bisacrylamide and polyacrylamide you use, you can get more or less porosity. If you use a high percentage, which is an example like this, a 20% gel, that means your pores are really, really, really small. And if you use a 4% gel, that means you've got very little polymer and hence your pores will be bigger. You can kind of think of it this way, right? When you have high percentage gel, that means your pore size goes down. So if you want to separate out large proteins, you want a low percentage gel. So here I'm writing down that when you have a high percentage gel, you reduce your pore size and you reduce the molecular weight of proteins that can pass. That's the idea behind porosity. We also should cover what it means to have polyacrylamide gels versus agarose gels here. So previously I talked about using agarose for gel electrophoresis. and agarose tends to have really large pores. Polyacrylamide, on the other hand, has much smaller pores. In order to reach the same pore size as agarose, you would have to have very, very little polymer in solution. And in that sense, you might actually not even be able to polymerize your polyacrylamide to reach the pore size that agarose has. We had covered that here. Remember that if you increase your gel percentage, then you reduce your pore size, which means you're adding more polymer and you're reducing the pore size. Or the flip side is you reduce your gel percentage by adding less of your polymer, and hence you increase your pore size. So that's why they've got agarose gel electrophoresis, which is meant for DNA because the pores are huge, the DNA samples are much larger, and polyacrylamide gel electrophoresis, which we're covering here, which has smaller pores and is designed for proteins. So I'll write that here. Proteins are really good in polyacrylamide, and DNA is really good in agarose gels. The other thing that they covered here was they had this concept of a 4 to 8% gel or a 4 to 20% gel to be able to separate out different sizes of proteins. What does that even mean? You can actually make gels with different percentages. So you could have a gel that has a fixed percentage, like here on the left, I've shown a 4% gel. If you take a cross section at the top and a cross section at the bottom of the gel, they'll look pretty much the same. They've got large pores, which are relevant to like a 4% gel. Now, if you have a 4 to 20% gel, what this means is that at the top, it's actually a larger pore size, and at the bottom, you've got a smaller pore size. And remember that if your pore size is big, then that means you can separate out proteins that have much larger molecular weight. So large molecular weight proteins will flow through these larger pores, but they won't really flow through these smaller pores that are here. So by using what we call a gradient gel, such as a 4 to 20% gel, you can actually separate out a larger variety of samples on the same gel, as opposed to a fixed percentage gel. The flip side is making a 4 to 20% gel is a lot harder, so you typically just buy it from a vendor, such as Bio-Rad or Rockland or Thermo Fisher. But the 4% gel, you can generally just make that in lab. It just has less resolution for your proteins. So hopefully you understand this concept. Your first step is to determine your porosity depending on the kinds of proteins that you're separating. And these guys have given a really good recommendation of the different protein sizes that you can separate on different percentage gels. We also covered the idea behind porosity. Then we covered how agarose and polyacrylamide are different. Agarose has larger pores, which is good for DNA. Polyacrylamide has smaller pores, which is good for proteins. And then we covered what does it mean to have a gradient gel versus a static percentage gel, and what are the advantages of each. All right, so our next step is to place the gel in the gel apparatus. This changes depending on which kind of apparatus you use. Here on the right side, we've got the Bio-Rad apparatus, but you might be using a different one, so I can't really discuss the details of the apparatus. In general, this is what it looks like. We've got some place to put buffer, some place to put a gel. We've got electrodes here, which will then get connected to the running apparatus, which will actually apply the electric field. And in the Bio-Rad case, you can actually put two different gels, one here and one here. So you can actually run two gels at the same time. Totally depends on the apparatus that you're using. Our next two steps are to prepare the SDS PAGE running buffer and also to fill the inner portion between the gel holders with appropriate buffer, as well as the filling of the outer chamber of our running apparatus. This is a lot, and it really depends on how the apparatus is built. But let me cover the theory behind it. So on the left, you'll see that we've got a gel that's surrounded by buffer. Why do we need this buffer? Remember that electrophoresis runs based on the principle of having an electric field. In order to have an electric field, you need to have a complete circuit. So the buffer allows you to have a complete circuit around the gel. That allows you to have the negative electrode or the cathode on the top of the gel as well as the anode on the bottom of the gel. And this makes it so that your samples actually go through the gel towards the positive side, as long as your samples are negative. And in SDS PAGE's case, all your samples should be negatively charged. We'll cover why in the next slide. Also on the right side, we see that same gel running apparatus. And what they mean by filling the inner portion and the outer portion of running buffer is here. Just take a look here. This is the gel. Here is the outer portion. So we're going to fill this chamber with a lot of running buffer. As well as inside this gel running chamber, you'll have to fill a little bit of running buffer here as well. This allows you to connect the top of the gel with the bottom of the gel in one electric field using buffer. Alright guys, you've made it through half the video. I know it's been a lot so far. We've looked at how do you actually prep the gels, what is porosity, how do you actually put them inside an apparatus, why do you even have sample buffer. It's a lot of information there. And then what's coming is how do you prepare your samples? How do you actually load them onto the gel? And how do you run the gel? I'd really appreciate if you liked or shared or subscribed or did all of the above so that you show some support for this channel. Because I'd love to know that my viewers actually like the content that I'm putting up. Thank you very much. Let's keep going. All right, our next step is to prepare a control lysate. Why do you need a control lysate? Because you want to make sure that you know that your gel ran properly. And by having a control lysate, you have at least one sample that you know always runs, and you can load some other unknown samples which may or may not run properly on your gel. To prepare the control lysate, we take pre-prepared lysate, and then we already have it in sample buffer. We thaw it, we add it into a microfiche tube, and then we add some beta mercapto ethanol or BME to it. And that's shown here in the protocol. Why do you need beta mercapto ethanol? It's because the proteins that are inside your prepared lysate have a lot of these disulfide bridges. And we want to reduce these because you don't want your protein to have a very complex tertiary or secondary structure. Ideally, what you'd like to have is, instead of having this kind of a complex protein going through your gel, you want something that's kind of a linear protein. And the reason for this is because if it's a complex protein and it's going through a pore, you're not sure that it's fitting through because the protein structure allows it to, or because it's actually the right size. But if you have it linearly, then of course you know that a linear protein can fit through that pore. Another way to think about it is proteins come in all kinds of shapes and sizes, right? So even if you have one protein that's say 100 kilodalton, this protein can look like that, it can look like that, it can look like that. So even though it's the same 100 kilodalton chain, it can have all kinds of shapes. And we don't know that as the shape is going through a pore, whether it's going through because it just fits through the pore because of its shape like this, or it's because it's actually the right size. So we linearize all the proteins that are in our solution. And then once we actually run them through the gel, the linearized proteins will separate according to their size only and not according to their secondary or tertiary shape. So that's why we add beta mercapto ethanol first to our control lysate to get rid of those disulfide bridges and reduce some of the secondary and tertiary structure. We'll be doing more of that later. All right, it's now time to prepare unknown samples. Like we did previously for our known lysates, we actually take our sample and we mix it with beta mercapto ethanol. But before that, we actually have to mix it with sample buffer. What is that? Sample buffer has our SDS, which is the namesake of SDS PAGE. And SDS is shown here. It's a really long chain along with a negative charge because of the sulfate and a positive counterion because of the sodium. When you add this to your proteins, it takes a protein that has a lot of charges and a lot of charge-charge interactions, and it linearizes it like it's shown here. So now, SDS also gives the protein a negative charge because of this sulfate. Remember that when you're running SDS PAGE, you need your proteins to have a charge, negative hopefully, and then those proteins will migrate towards the positive anode. SDS PAGE absolutely requires you to have SDS to impart that charge to a protein and to linearize it, removing any secondary and tertiary structure by using beta mercapto ethanol and SDS. The other thing that they ask you to do in this protocol is to prepare some molecular weight standards. I talked about this in the Gel Apparatus YouTube video, so I'm not going to really cover it in detail, but in general, a molecular weight standard allows you to make sure that your proteins separated, and it tells you the size of the proteins that's separated when you do your analysis later on. They have a note here saying that depending on what your downstream application is, whether it's Western blotting or not, you should choose the appropriate standard. I don't want to cover that here, but make sure that you choose the right protein ladder depending on what you're doing downstream. And in the case of doing Western blotting, use pre-stained molecular weight markers. Another good thing about having sample buffer is not just that it has SDS and it has the beta mercapto ethanol, but it allows you to visualize your samples. So here you can see a student that's loading a gel. And in the gel, they've got these little blue samples that's shown here. Protein by itself is invisible to the naked eye. So what we need to do is to color it, and at least color our samples so we can see them while we're loading them. Plus, when we run them, the sample will actually separate out. So initially you might have it here, and over time you'll kind of see a dye front while you're running the gel, which tells you how close the gel is to completion. We'll look at that in the next section. The other thing I wanted to show you was here's what a ladder allows you to do. Both of these lanes actually contain ladders. And here you can see our samples in the middle. So by looking at the ladder, you can kind of say, hey, if a 34 kilodalton protein is here, maybe this sample is a little bit larger than 34 kilodaltons, but a little bit smaller than 43 kilodaltons. So maybe it's something like 38 or 35 kilodaltons, in that range. So that's what a molecular weight marker allows you to do. It allows you to tell your protein size when you're doing your analysis downstream. Our next step is pretty simple. Take our samples that are inside microfuge tubes that already have SDS and beta mercapto ethanol and boil them for 5 minutes. After the 5 minutes, we spin them down, and what we should see is some kind of pellet, and our rest of the sample is inside our supernatant. The reason we see a pellet is because beta mercapto ethanol and SDS will also cause other parts of the cell to break down inside our unknown samples. And any of this junk, that cell debris, we want to get rid of it from the gel because it'll mess up our gel's resolution and how nice our gel looks. All right, now it's time to load our samples into the wells. This is pretty straightforward. All you have to do is use these tips that are designed to load gels inside the gel apparatus. And the things to keep in mind are how much volume are you actually loading into each well, as well as how much protein are you actually loading inside each well. Make sure that you don't overload the amount of liquid, because that will cause you to load maybe some liquid here, and as you're squirting out the liquid, some of it will start transferring over to the well next to it. That would be a problem. The other thing is the amount of protein that you load. If you load too little protein, you won't be able to visualize it downstream. Of course, it depends on the techniques you use. And if you load too much protein, the visualization will be really tough because you might oversaturate your signal. So just make sure that you follow some of these guidelines. What they're saying is about 1 microgram of purified protein is sufficient for visualization. This is generally your lysate or purified protein that's like recombinant that you just bought. And then 10 micrograms is sufficient to visualize proteins inside a lysate or a Coomassie stain gel. And Coomassie is a particular downstream method that we could use to visualize our samples. Otherwise, you might also think of Western blots, which of course will have a different amount of sample that you'll be loading. So make sure that all this complies downstream. All right, it's now time to run our gel. This is pretty much the last step in this tutorial. I want to be covering a little bit about the downstream kinds of analyses that we could do, but let's just say that this is the last step of SDS PAGE. In this step, all we do is we connect the apparatus so that the electric field is complete and the entire circuit is complete. And then we click start. Here what this suggests is running it at 150 volts. This really depends on the kind of gel that you use. Some gels can actually run at a higher voltage and they can run faster. So your voltage determines your speed. I'm going to write that here. Higher voltage means higher speed and less duration for your overall SDS PAGE run. Here, what they've suggested is running for 45 to 90 minutes and then turning off the power as soon as the dye front migrates out of the bottom of the gel. Do you remember what the dye front was? The dye front is essentially what happens when you add your sample buffer to your samples. So you get this blue kind of solution. And that blue will actually run faster than your protein because it's a very, very small dye. Once that small dye runs off the gel, you might end up seeing something like this. So you might have your gel, you'll have your samples, but you can't really see them. They're still running. And at the bottom, there'll be this blue color that will eventually reach really close to the bottom, which is when you stop it. So this is what they're suggesting. Stop it as soon as the dye front migrates out of the gel or reaches really close to the bottom of the gel. On the image on the left, there's a new concept I wanted to introduce to you. Remember that we were running a 4 to 20% or 4 to 8% gel. There's also a possibility that you might run the traditional form of SDS PAGE, which has what we call a stacking and a resolving gel. A stacking gel is a gel that has a lower percentage, and hence any sample can migrate through it. So you can see on here, there's a sample that we put inside the well, and as it migrates, it goes down and further down, and then it reaches this boundary between the stacking and the resolving gel. The resolving gel tends to be a higher percentage. And that means that as soon as these proteins reach the interface between the low percentage and the high percentage gel, then they end up squishing together, kind of like hitting a wall, but the wall is somewhat permeable. So they squish together and then they start separating, which is what you see here. And as they separate, now they actually resolve inside the resolving gel. The other thing that you see is you see some ions here. These ions are actually what's in your running buffer that help push your proteins through the gel using the electric field. And the last thing I wanted to cover within this YouTube video was the idea of post-run analysis. So proteins in general aren't really easy to visualize because, well, you can't see them with your naked eye. So you need some kind of a stain or some kind of a way to visualize them inside your gel. Two methods that are pretty common are Coomassie staining and Western blotting. In a Coomassie stain, I've provided you a protocol here. I won't really go through it, but in general what you do is you take your gel, you put it inside the Coomassie stain, you heat it for some time. Here you can see there's a microwave and a gentle shake. And then afterwards, you prepare a destaining solution and you destain whatever didn't bind to your protein. So Coomassie is a stain that will bind to your protein. It'll give it a color, but then it also won't bind to anything else. So you can just destain and remove it. And the end product kind of looks like this. So you can see it looks like they ran a ladder here, maybe some known lysates here, and then these are maybe their unknowns. So it's a nice way of visualizing your protein. Another method is called Western blotting. And here you only see one band. Why is that? Western blotting is a method to analyze one single protein by using antibodies. It's way more sensitive than Coomassie. It doesn't visualize the entire gel and allows you to analyze a single protein, which is probably the proteins that you're interested in. We'll cover Western blotting in a future video. All right, with that, we're all done with our SDS PAGE step-by-step tutorial. I really hope that you enjoyed it. I hope you like and subscribe to this channel, and you visit SciGine.com to be able to search through our database of biology methods and find an SDS PAGE protocol that's specific for the proteins of interest that you have. Thank you very much. Talk to you later. This is Carter, signing off.